By cell count, you are approximately half human. You share your body with 38 trillion bacterial cells, an ecosystem equal to your own human cells. For most of medical history, science treated these microorganisms as passive digestive passengers. We now understand they act as biological co-pilots, influencing the system from the inside out. These microbes manufacture 95% of the body's serotonin and significant amounts of dopamine. They are the primary factory for the neurotransmitters that govern your mood and reward systems. This challenges our basic sense of autonomy. It suggests that your daily thoughts and baseline moods are being heavily influenced by a hidden world you did not consciously choose. The enteric nervous system is a dedicated hardware network, a fully independent mesh of 500 million neurons in the gut, exceeding the entire spinal cord. It communicates with the brainstem through the vagus nerve, a major neural highway bridging both systems. The sheer volume of this communication reveals a startling hierarchy. Between 80 and 90% of the signals traveling along the vagus nerve are moving from the gut up toward the brain. In this biological arrangement, the brain functions largely as a receiver. It is constantly processing a massive, non-stop stream of complex commands and status reports originated entirely by the gut. This upward signaling is written in the chemical software of the mind. Gut bacteria synthesize the molecules required for human emotion, specifically serotonin for mood regulation, dopamine for reward, and GABA for stress response. Standard antidepressants are designed to increase serotonin availability in the brain, yet 95% of that serotonin is built in the gut by bacteria. Your capacity to feel reward or maintain a calm baseline depends on the availability of these microbial precursors. If the bacteria are missing, the brain physically lacks the materials it needs to regulate your mental state. Focusing therapy solely on the brain ignores the biological factory floor where the actual chemicals of human emotion are manufactured. In April 2026, researchers at Harvard Medical School identified a specific molecular pathway that links gut bacteria directly to clinical depression. They discovered that the bacterium Morganella morgani interacts with environmental pollutants to produce a specific phospholipid. This molecule triggers neuroinflammation, a condition strongly associated with depressive symptoms. This mechanism is supported by 2019 European data showing that people with depression consistently lack two specific bacterial species involved in dopamine production. This has led to the use of fecal microbiota transplants, where transferring bacteria from healthy donors into depressed patients has resulted in complete symptom remission. When physically transferring an ecosystem from one person to another cures a psychiatric illness, depression reveals itself as an ecological failure rather than a localized brain defect. Research from Northwestern University in 2026 indicates that gut microbes influence the foundational physical structure of the brain during early development. Scientists transferred microbiomes from various primate species into young mice and observed that the mice's brain development shifted to resemble the donor species, matching their specific gene expression patterns. The experiment produced startling results. Mice that received microbiomes from smaller-brained primates developed neural patterns mirroring ADHD, schizophrenia, and autism. This provides evidence that the gut microbiome is shaping brain function during development. If a child is not exposed to the right microbial colonies in early life, the brain's foundational architecture may develop differently, potentially leading to neurodevelopmental symptoms. This chart compares the growth of human brain volume with the increasing density of the human gut microbiome over millennia. Our species' intelligence likely required a symbiotic partnership to fuel the massive energy demands of the cerebral cortex. Without these microbial partners providing metabolic support and specific neurosignaling, higher cognition would physically fail. Human intelligence is not a solitary evolutionary achievement, but a co-authored success story dependent on the organisms living inside us. This ancient biological partnership is now facing a crisis. C-section births, antibiotic overuse, and ultra-processed diets are rapidly eliminating the critical bacteria that have supported human health for thousands of years. The absence of foundational microbes, like CAG 170, leaves the human host vulnerable to a wide range of chronic physical and mental health conditions. We are currently observing what happens to the stability of the human mind when its ancient biological support system is starved and sanitized within a single century. This process is happening right now in your immediate reality. Millions of microbial signals have already dictated your baseline anxiety, your hunger, and your mood today. While we do not choose the colonies that inhabit us, they dictate our mental patterns without our conscious consent. If you are not the sole author of your thoughts, how do you redefine your sense of self?